Hey everybody, it's Dr. Gilchrist, and welcome to week 3 of our journey into drugs and behavior. So this week we are going to talk about opioids, we're going to talk about psychomotor stimulants, and we're going to talk about nicotine and caffeine. Just as a friendly reminder, this week you have your first writing assignment due, as well as your standard 2 forum posts and your third quiz. But let's go ahead and begin by discussing opioids. So I'm going to give you a brief overview. We're going to start with a brief history of opioids. We're then going to talk about opioid receptors and endogenous neuropeptides, better known to you as endorphins, although endorphins are just one type of endogenous opioid. We'll talk a little bit about the primary role that opioids often tend to play, which is pain relief. We'll talk about tolerance, dependence, and withdrawal upon opioids. And then we'll talk about opioid addiction, and we'll talk about different methods for treatment. Now, we are kind of jumping around at this point. Previously, we were in other chapters for talking about alcohol. We were in chapter 8. Now we are jumping around to chapter 10. And you might be asking yourself a little bit, why did I kind of jump around from alcohol to opioids in this particular order? I wanted to start by kind of looking at the depressants before moving on to different classes of drugs, including stimulants. So let's start by talking about a very brief history of opioids. So what are opioids? So opioids are technically a class of narcotic analgesics. What this basically means is that they are designed to reduce pain. That's what the term analgesia means. And they're going to do so without potentially causing unconsciousness like barbiturates or heavy tranquilizers might. Now, opioids tend to have some common effects. I mentioned that they are pain relievers. They also exert depressant effects on the body, very similar to alcohol. So some very common effects will include relaxation, promoting sleep. However, at higher doses, coma and death can result. And oftentimes this will happen because, much like alcohol, this will happen because of respiratory depression. Basically, the depressant effects of the drug become so heavy that it is no longer possible to breathe in these cases. So opioids there are a variety of different types of opioids that you have probably heard about. And so I wanted to kind of share with you some of the different DEA schedules for different types of opioids. So buprenorphine, which we will talk about as a partial agonist for different types of opioids, is Schedule 3. Codeine, which you may have had as a cough syrup preparation, is a Schedule 5. Diacetylmorphine, known as heroin, is a Schedule 1. Desomorphine is a Schedule 1. Fentanyl, which I'm sure you have heard a lot about, is a Schedule 2, as is hydrocodone, morphine, methadone, opium extract, and thebaine. So we recognize that opioids do have a variety of different therapeutic purposes. And so because of that, those lower grade opioids are often going to be at lower schedules. In addition to those opioids, you may have heard of a drug called dextromethorphan, which is present in a lot of cough syrups. That is a low-grade opioid that is even kind of less potent than codeine, so it's sold over the counter in cough syrups, although I do believe here you have to be over 18 to buy them. Another example of an opioid that you might have heard of that you buy over the counter is loperamide, better sold under the brand name Imodium, which is often used to basically stop diarrhea by slowing down the digestive tract. Both of those are low-grade opioids that you can actually purchase over the counter, provided that you're over 18. So where does opium come from? So we do have some that are produced naturally in the environment, and as we're going to talk about later, these can also be fully synthetic or semi-synthetic in nature. Opium comes from the extract of the poppy plant, Papaver somniferum. It's a flowering plant. As you can kind of see here, you don't see the flowers. It basically stands about 3 to 4 feet high. It tends to have big flowers. You can see that there's a seed pod right here. And basically, the flower petals will fall off. You're going to score the pod with a knife, much like has been done here, and that substance is going to basically come out. This is raw opium. And basically, it's going to be scraped from the seed pod. It's going to be either used as is, or it is going to be further refined and processed. So most opium cultivation, particularly those that are illegally produced, occur in Afghanistan. However, opium also comes from places like Pakistan, Myanmar, Colombia, and Mexico. And these have been used recreationally and medicinally for thousands of years. But I want to talk about how different opium or opioid byproducts ended up kind of becoming more common. And one of those ways particularly occurred in the form of laudanum. So basically what you're going to do with laudanum, you're going to take some opium, dissolve it in an alcohol So basically you're going to take a tincture of opium and dissolve it in alcohol. And you're basically going to produce laudanum. And additionally, people would often put laudanum in their wine, and you could see, and it was very acceptable both in Victorian England and the United States. This was a very common medicinal treatment, particularly for things like cough. One of the things that we know about a variety of different opioids is that they open up the respiratory passages and they make it easier to breathe. It's part of the reason why codeine is in cough syrups are often going to be used for very bad coughs, because those opioids, through their depressant effects, are going to open up those respiratory passages. I do find it kind of interesting. Always useful. Paregoric is another type of opium preparation. Always useful, both for children and adults. Yeah, probably not. Now, morphine is going to be the active ingredient that is found in naturally occurring opium. This was isolated in the 1800s, and this was actually a very leading finding in psychopharmacology, because it was really the first time that an active ingredient was isolated from a plant. So this was a huge deal. And because you could isolate it at that point, that meant that doctors could begin to prescribe different dosages for people. It wasn't just, just take this plant, it'll be fine. At this point, you could start dosing, and that was helpful. You didn't want people to overdose if they didn't need to. Now, there are some additional compounds that are in opium besides morphine. So codeine is, again, a naturally occurring substance, as is thebaine and narcotine. Now, that is not to be confused with nicotine. Nicotine is a stimulant. It is a completely different substance. Narcotine is an opioid compound, and so this is not a typo. Now, these and morphine are going to have slightly different chemical structures, and as a result, this is going to basically determine the effectiveness as well as some of the different side effects that may occur. So here I'm actually showing you the chemical structure for morphine, codeine, heroin, and naloxone, which is an antagonist of opioids, but is itself an opioid. So one of the things that we're going to talk about, morphine and codeine are both naturally occurring. You can see there are some slight differences here. Heroin looks very much like morphine, but it has 2 acetyl groups that you can see right here that make the drug more lipid soluble. It can pass through fats more easily, and as a result, it is much more potent and it enters the brain more easily than morphine will. Naloxone, as an opioid, also has a similar chemical structure, but this addition actually makes it antagonistic to the actions of opioids like morphine and heroin, or OxyContin. Now, what about heroin? As I kind of mentioned before, heroin, as you saw, has a very similar chemical structure to morphine, but it has two acetyl groups added to that morphine chemical. As a result, it makes it more lipid soluble, it reaches the brain more quickly, and as a result, it is often going to be more potent than morphine. Now, one of the things you may not know is that once heroin reaches the brain, it is then converted into morphine. Now, modifying morphine or the other opioid compounds basically create other types of opioids. So for example, we can create what are known as partial agonists. So they bind to opioid receptors. They generally do enhance somewhat the effects of the neurotransmitters. However, they're going to have fewer biological effects because they're not perfect agonists. And additionally, you can create what is known as a pure antagonist. These are structurally similar, but they have no efficacy. That basically means that they're not going to cause any neurotransmitter effects. They're basically there to block or reverse opioid binding. So going back to here with our chemical structure, that's exactly what naloxone is. For those who don't know, naloxone is sold under the brand name Narcan. So if somebody has overdosed because of respiratory depression, naloxone or Narcan basically is immediately inserted into the system, into the body, and when it's in the body, it is basically going to bind to those opioid receptors so that the drug has nothing to bind to. It will also clear out and reverse opioid binding from those receptors so that those overdose effects can stop and the person can breathe again. So Narcan can be applied in cases of overdose, but it has to be applied very quickly. Now, let's talk a little bit about fully synthetic and semi-synthetic opioids. So when we talk about semi-synthetic opioids, we're basically talking about drugs that are synthesized from naturally occurring opioids. So that includes diacetylmorphine, aka heroin, desomorphine, which is known as crocodile. So if you've ever heard about a heroin or a drug that's injected that basically eats flesh, that would be crocodile. Do not look it up if you have a sensitive stomach. Buprenorphine, which we'll talk about later, hydrocodone, oxycodone, and that would include any variations that include oxycodone, like Percocet, which is basically oxycodone and acetaminophen. Then we have our fully synthetic opioids. These are not derived from naturally occurring opioids, so they are fully synthetic. This will include fentanyl, methadone, and levetiracetam. So here's kind of another way of looking at this. So we have our opium, which contains things like morphine, codeine, and thebane. Here we have our semi-synthetics, including heroin, hydromorphone, better known as Dilaudid, oxycodone, which is also known as Percodan, OxyContin, etc., atorphine. And then our total synthetics, that includes penazocine, meropenem, fentanyl, methadone, LAAM, which I just showed you here, so that's LAAM, and then propoxyphene or Darvon. And these have some similarities to our endogenous opioids, including enkephalins, endorphins, dynorphins, and endomorphins. Now, what are some common routes of administration for your opioids? So for things like morphine, often this is going to be used as a pain relief process or in hospice care. This may involve an intramuscular injection or an oral administration with a swab. With opium, this is often going to be inhaled via smoking. With heroin, that can include snorting heroin powder, subcutaneous IV, subcutaneous injection, which is sometimes referred to as skin popping, or IV injection, which is best known as mainlining. And with IV injection, heroin tends to reach the brain in about 14 seconds. This is going to become really important when we talk about nicotine later on this week. Now, talking a little bit about the pharmacokinetic properties of opioids. So, there is going to be some variability in how quickly drugs pass into the brain. So, for example, this is kind of based on lipid solubility. So generally, the more lipid soluble an opioid is, the more quickly it's going to pass through tissues and reach the brain. So heroin is, as you can see here, is going to cross through that blood-brain barrier much faster than morphine does. And because you get that faster action, it's often going to be more reinforcing than morphine is. Like many drugs, opioids are going to be metabolized into the liver. So you're going to get those metabolites as a result. So for example, your textbook notes that morphine produces a metabolite called morphine 6-glucuronide, which is a pain reliever that can potentially lead to morphine's analgesic effects. So morphine is also a metabolite of codeine. It's a secondary metabolite of heroin. So those metabolites can still have psychoactive properties on them. Now, elimination rates are going to kind of vary. It depends on how many things need to be metabolized. Generally, what we're going to find that we'll talk a little bit more about later, if you're struggling with heroin addiction or OxyContin addiction, oftentimes one of the things that's very common is what is referred to as methadone maintenance. Now remember, methadone is also an opioid. It is a synthetic opioid, but it's often going to be used to basically help people who are struggling with those opioid addiction. Methadone tends to be slower acting than something like heroin is. So oftentimes what we're going to find is that those methadone levels peak very late after administration. And basically, it's there to prevent heroin withdrawal symptoms from occurring because methadone is also an opioid. So there is some cross tolerance there. As a result, you're going to feel less of those withdrawal effects. But it doesn't really produce a lot of positive effects, and it also takes a really long time to be cleared from your body as a result. So after reaching the bloodstream, how are opioids distributed throughout the body? So only a small amount of morphine is actually going to cross the blood-brain barrier. It is going to cross a placenta much more easily though. So again, if you're wanting to become pregnant, probably not a good idea to take opioids. For things like morphine or heroin or things that are faster acting, it's typically going to be excreted in the urine. It's generally going to take about 24 hours. Like I said, if we're talking about something like methadone, it's probably going to take more than a day. Now, let's talk about some of the major effects on the central nervous system and also the gastrointestinal tract, because those are the two major areas where we see a lot of opioid effects. So the central nervous system effects are obviously going to be related to dose and the rate of absorption. So the route of administration is going to matter, the type of opioid that you're taking matters, and the dose that you're taking matters. So in low to moderate doses, you will experience things like pain relief, constricted pupils. As an aside, if you want to figure out if somebody has overdosed, if you know somebody who may have overdosed from opioids, one of the quickest things you can do is check their pupils. If their pupils are constricted, they are under the influence of opioids. Things like drowsiness, poor concentration, dreamy sleep. But there are going to be some adverse effects, particularly at those higher doses. Dysphoria, so basically generally feeling bad, anxiety, nausea, and vomiting. And at higher doses, there can be unconsciousness and respiratory failure, which can lead to death. Now, as I kind of mentioned, loperamide is a low-grade opioid that is often used to treat things like diarrhea or gastrointestinal distress. People found out a while ago that opium and morphine do very similar things. Now remember, part of the reason for this is that opioids are depressants. That means they're going to slow gastrointestinal activity down. But we really don't want people kind of hooked on opium or morphine. Rather, we want to make sure that we're using a low-grade opioid that will become less problematic. And as a result, loperamide, often sold under the name Imodium, is more commonly used today. Now, one of the things that we are finding, though, is that with a lot of people addicted to opioids, when they go through withdrawal symptoms, it has been found that these people will buy mass quantities of loperamide. Another story that I have heard is that people buy mass quantities of loperamide so that they can basically get high. And so there has been some discussion about regulating, much like we do with pseudoephedrine for potential purposes of making meth. We may potentially start regulating how often people can buy loperamide so that people either aren't using it to manage withdrawal symptoms or are using it as a means to get high. Now, one of the things that we do find that is a major side effect of pain treatment with opioids is constipation. So a couple of years ago, I actually thought it was very interesting. It's one of the first times that I've seen this as a Super Bowl ad, but they ended up doing an advertisement for a medication that can be prescribed for opioid-induced constipation. Remember, this is a depressant. It is slowing down the motility and the movement of the gastrointestinal tract. So constipation is a very common thing that happens when you are undergoing pain treatment with opioids. So now let's move on and talk about the opioid receptors. So there are 4 different types, and we're going to talk about each of these. The mu opioid receptor is probably going to be the best known, but we have the mu opioid receptor, we have the delta opioid receptor, we have the kappa opioid receptor, and we have the nociceptor orphanin FQ receptor. Your textbook also refers to it as the ORL1 receptor, opioid receptor like 1 receptor. We're probably going to talk about the first 3 more. So your textbook calls it ORL1. I've always referred to it as NOPR, and it's a little easier to say, but each of these 4 receptor subtypes has a different distribution in the brain and in the spinal cord, and as a result, you are going to get a wide variety of physiological and behavioral effects. So here you are looking at the brain of a rat, and you can kind of see here the different concentrations of these different opioid receptors. So you can see that the mu opioid receptor tends to be concentrated more in areas like the striatum, the thalamus. You can even see a little bit of the locus coeruleus and the Raphe nuclei, which we talked about previously. For the delta receptor, we see a lot of action in the olfactory areas of the rat, as well as the striatum. And then with the kappa, we have a little bit of the striatum, the thalamus, parts of the cortex, and the locus coeruleus and the Raphe nuclei, though certainly not to the same extent as the mu receptor. So let's start by talking about the mu receptor. So the mu receptor is probably the best known opioid receptor. It has a very high affinity for morphine. It bonds binds with morphine very easily. And basically, locations of those receptors in the brain are going to reflect morphine's effects. So that includes analgesia, that pain relief, feeding, and the positive reinforcement that's produced, cardiovascular and respiratory responses. And because of that focus on the striatum, we also might have sensory motor integration. The delta receptor is largely going to be found in the forebrain, near the front of the brain. It's going to be involved in things like smell, motor integration, again, some of those reinforcing properties and those cognitive functions. And there is, as you kind of saw in the previous image, there is some overlap with the mu receptor locations. So there's possibly pain relieving properties in these receptors as well. And then we have the kappa receptor. And the kappa receptor, part of the reason that it has the name that it does is it's identified by binding to ketocyclosocine. And that's going to produce potentially hallucinations and dysphoria when you take this particular drug. And largely the kappa receptor is going to be found in the striatum, the amygdala, the hypothalamus, and the pituitary gland in humans. As a result, it potentially plays a role in pain perception, gut motility. So basically, is the gastrointestinal tract moving? Is it speeding up? Is it slowing down? And it potentially also produces dysphoria. Now, what about the NOPR? Well, the NOPR is basically found throughout the central and peripheral nervous systems. Areas that tend to have higher concentration include areas like the cortex, the limbic system, so areas like the amygdala, the hippocampus, the thalamus, the Raphe nuclei, and the spinal cord. And so the NOPR has been, again, kind of implicated for analgesia, learning, feeding, motor function, and endocrine regulation. Now, we don't know much about opioid receptors, but researchers believe that they are most likely metabotropic in nature. So the receptors are going to be linked to G proteins, and here is a proposed structure of the mu receptor as an example. You can see this is pretty complicated, so we're not really going to talk about this very much. So one major question that came up for researchers in the past: why do we have receptors for these drugs? So why do we have receptors for opium derivatives? And the researchers reasoned that if we have these receptors for these types of drugs, our bodies must be making similar natural versions of these opioids. So this would mean if we already have these receptors, that must mean that our body makes its own endogenous opioids. And by 1974, and I really want to put this into perspective, this was 50 years ago that we found this. 50 years ago, we were able to identify endorphins. Endorphins are peptides that bind to opioid receptors. And as I kind of mentioned, the fact that we call it endorphins is a little bit of a misnomer. Endorphins are just one type of peptide that we make endogenously, but it is not the only one. So endorphins are basically created by propeptides. Propeptides are going to be peptide precursors, and here they all are. So here we have pro-opioid melanocortin, or POMC. We have pro-enkephalin. We have pro-dynorphin, and we have pro-nociceptin orphanin FQ. And basically cleaving these different propeptides creates different peptide products. So for example, proenkephalin can be cleaved to form here metenkephalin or leuenkephalin. And you can see some of the other peptide products that can be created based on this cleavage. So for example, if we cleave this portion of POMC, we end up with beta endorphin. So propeptides are basically found in the brain, the spinal cord, and the autonomic nervous system. And these are going to be concentrated in areas that are related not only to pain, but also to mood. So now let's talk a little bit about pain perception and how opioids try to block that pain perception. So if we want to understand the effect of opioids on pain, and let's be very clear, most people who are taking opioids are probably taking them for pain relieving properties. So if we want to understand the effects that opioids have on pain, we actually have to understand a little bit about how pain actually works. So pain can be caused by a variety of different types of stimuli. And that's because you and I have many different nociceptors, which are especially responsive to pain, embedded throughout our body. And pain can vary in terms of its intensity, its quality, and its subjective perception. We can have a pain that's very sharp and very fast, such as when we get a paper cut. Or when we stub our toe, we may experience a little dull throb for a longer period of time throughout the day. So pain can vary in a variety of different ways. And our understanding and our sense of pain can be modified by a variety of different things. It can be modified by our emotion. If we're already scared of getting an injection at the doctor, our fear is going to make the injection feel worse. It's part of the reason why when you're a kid, they try to distract you, because that makes the shot feel less bad. Opioids can also modify that pain. Things like acupuncture can modify that sense of pain. And I want to talk a little bit about two major components of pain. We have what is called the early or the first pain. This is your immediate sensory component to pain. So if I stub my toe or I drop something on my toe, that very specific localized spot is my first pain. It's going to be limited to the area that experienced some kind of damage. And in particular, this is very fast acting, and this is because those fibers, which you can see here in blue, are due to myelinated A delta fibers. So if you can with me, we'll get pain. So let's say I stub my toe. That localized pain is going to be present in the lumbar, enter into the lumbar spine portion of the spinal cord. It's going to travel up to the medulla, then to the midbrain, and then finally, it is going to be processed in the thalamus and then in regions of the somatosensory cortex. But I also want to talk about that late pain. So initially, I stub my toe and it hurts. But over time, I feel a different type of pain. Whereas the first pain, the early pain, is very sharp, immediate, and localized, late pain or second pain has often been argued to be more of the emotional component. So whereas when I initially stub my toe, I feel it right in the area where I damaged it, later on I may feel a dull, throbbing ache throughout my whole toe. It's less localized to the area of damage. And this late pain has been proposed to be due to slower C fibers. So the C fibers are here in red, and we're going to see a little bit about what they do. So the C fibers, again, are going to enter in that region of spinal cord, travel up through the medulla into the midbrain. They're going to make contact with the periaqueductal gray, which we'll talk about later, as well as parts of the reticular formation, before going into the thalamus and again, having some processing in the somatosensory cortex, but also in areas like the insula. And we have some evidence. So these are two different areas that are being processed, giving evidence for two different types of pain. And we have some additional evidence with neuroimaging. So participants get exposed to some type of painful stimulus, and then their pain rating is monitored during an MEG. So here we're looking at changes related to muscle movements. So here we're going to start here. So here you can see that initial early pain. You can see that it's very short-lived, it's very localized, but the late pain hasn't really begun yet. But over time, what we can see is that the early pain signals are decreasing, but we're getting this increased late pain, which continues to grow past the point of the early maximal pain to the point where the late pain is then recorded. Now, if we map this onto fMRI data, here's what we tend to find. Early pain is largely going to activate primary somatosensory cortex in the contralateral region. So if I damage my right hand, this is going to be in my left somatosensory cortex. Both early and late pain activate the secondary somatosensory cortex, and they do so at a bilateral level. And then late pain activates the anterior cingulate cortex, and it's been suggested that it's the anterior cingulate cortex that is responsible for pain's emotional component. So how do opioids factor into all of this? So opioid receptors are going to be located throughout your body. And there are three mechanisms of how opioids potentially work that we're going to talk about in greater detail. First of all, they can act upon inhibitory interneurons in the spinal cord. Second, they can affect descending pathways in the periaqueductal gray. Or third, they can affect higher order brain sites like the cortex. So let's talk about each of these in a little bit more detail. So we know that opioids can potentially act on spinal interneurons. So here we have a nociceptor in the sensory system that is going to eventually be projected into the brain. Here we have a descending inhibitory pathway. Now, let's not worry about the pathway right now. I want you to kind of focus on this part right here. So basically, you're going to get pain. That free nerve ending is basically going to be sent to a neuron that projects to the brain and the spinal cord. So if it feels pain, it's going to activate that projection neuron. But inhibitory interneurons can actually release endorphins that inhibit these projection sites. And morphine and similar opioids can ultimately bind to these receptors. So basically what we're doing is we're inhibiting this projection, and we're basically blocking pain transmission at the level of the spinal cord to those higher order areas. Now, additionally, opioids can modify signals from our brain. So descending pathways can, as you see here, inhibit spinal cord pain transmission. So here we have our nerve fiber. It's going to send information to an excitatory interneuron, and that is going to basically send that pain information through a projection neuron. We have these modulatory pathways that can modify the interneuron signals. So we can inhibit the projection neurons just like we did here, or we can inhibit these excitatory interneurons. So that's another way that we can kind of block that pain signal. We can basically say this nerve in the spinal cord is feeling pain, but we're basically going to inhibit this interneuron so that no pain information is sent to the projection neuron. Additionally, what we can also do is excite inhibitory opioid neurons. So this pathway can excite the opioid neuron, which inhibits that pain message to be sent to the projection neuron. Now, I want to talk a little bit more about these different pathways. So we have a modulatory pathway, which can inhibit, and we have another one, which is excitatory, but can act on opioid neurons that inhibit. The origin of these inhibitory pathways is here in the periaqueductal gray of the midbrain. And researchers have actually found if you stimulate the periaqueductal gray, that can actually treat chronic pain. And as you're kind of looking at these descending pathways, here's what I want you to notice. And you can kind of follow my arrow here. So in this descending pathway, notice that there are two different areas that are being made contact with. So first of all, we have contact with the locus coeruleus, and then in areas kind of related to the reticular formation, we have contact with the Raphe nuclei. So these are two additional neurotransmitter areas that might get involved with these descending pathways. Remember that the locus coeruleus is a source for many noradrenergic neurons, that is, neurons that release norepinephrine. The Raphe nuclei are the source of many serotonin-based cells. And then finally, opioids can actually modify higher order brain centers. So pain, as we kind of mentioned, has an emotional component, and that is particularly true for areas like the anterior cingulate cortex. In addition to this, the higher order brain areas can also affect autonomic nervous system responses and neuroendocrine responses. We actually find that there's a negative correlation between mu receptor activity and reported pain scores, and the areas that seem to be really critically involved in this include the nucleus accumbens, the amygdala, and the thalamus. So now let's talk about reinforcement, dependence, and tolerance. So one of the things that you need to know is that in general, opioids are incredibly reinforcing. There's a lot of research from animal studies that shows this. So what you're looking at here is a setup for electrical self-stimulation. So basically, an electrode is placed in an area like the nucleus accumbens, and basically every time the rat presses the bar, they get a nice little stimulation in the nucleus accumbens. And what we find is that if you apply opioids to these animals, opioids actually lower that threshold for stimulation. So it takes less stimulation to basically produce that reward. So opioids do tend to enhance those reward mechanisms. Now in human beings, this is going to be a little different. We're talking about the subjective effects. What we do tend to find is that most people will experience a rush, a high, a nod, and a straight. So the rush is the initial rapid onset of euphoria. For a lot of people, this is going to be really reinforcing. Then we have the high. People feel very joyful. They feel very relaxed. They feel at ease. Then we get the nod. This is when you're basically going from that euphoria and positive feeling into a sense of calm, disinterest. You might even sleep a little bit, hence the nod. And then finally, you have the straight. And this is basically the period between craving an opioid and then feeling the positive effects. So it's basically kind of those feelings of normalcy. And we do find in animals that self-administer, they do create a stable blood level of morphine, kind of indicating that, again, opioids provide a lot of reinforcing effects. So as I kind of mentioned, here are those subjective effects. So we have our rush, our high, our nod, and our straight. Now, part of these reinforcing effects are due to a couple of different things. We know that opioid use is associated with changes in both GABA to kind of get those inhibitory and relaxing effects, as well as dopamine. And your book talks about this a little bit more. One of the things that we do find is that opioids very easily pair with environmental stimuli. So they've kind of looked at this with rats in what is called conditioned place preference. So basically the idea here is that animals basically create an association between a particular environment and a drug's reinforcing effects. So basically there might be a shuttle box with either a drug in one or a vehicle, which is basically saline. It's basically meant to look like the drug, but it's basically a placebo. And what they tend to find is that After presenting the rat with the option for either the vehicle or the drug, rats will often return to the place where they received the drug. And so we do actually find conditioned place preference for morphine. You can check box 10.1, that corresponds with page 320 in your book, to learn a little bit more about it. Your book also talks about the fact that NMDA receptors may also be involved with this, particularly with that conditioned place preference. Now, I mentioned dopamine, and this is where the mesolimbic pathway really starts to get involved here. Just as a reminder for you, the mesolimbic pathway is made up of, it starts in the ventral tegmental area of the midbrain, and it terminates in the limbic system, particularly in the nucleus accumbens. And what we basically know is that when opioids reach the ventral tegmental area, that increases firing of those dopaminergic cells. As a result, we get an increased release of dopamine into the nucleus accumbens. We get those positive reinforcing effects. We get that sense of reward, and it's very easy to become addicted. But one of the things that you need to be careful with is that not all opioids produce this increased dopaminergic firing in the mesolimbic pathway. We do actually find that if you stimulate a kappa receptor, you actually get reduced dopamine activity. So how is ventral tegmental cell firing increased by opioids? So here is the ventral tegmental area, and here is a GABAergic cell. So here's beta endorphin. Beta endorphin, now remember that the job of GABA is to be inhibitory. So what you can kind of see here, GABA is going to send an inhibitory message to this mesolimbic cell, which is going to reduce dopamine release onto the nucleus accumbens. Now, so GABA is going to be inhibitory, you're going to get less dopamine. Basically, what beta endorphin and opioids do is they're going to inhibit those GABAergic cells. So basically, we are canceling these two out. By inhibiting GABA, the ventral tegmental area cannot reduce dopamine. As a result, we get increased firing and greater dopamine release into the nucleus accumbens. Now, dynorphin, which is another opioid, and it's one of our endogenous opioids, dynorphin actually works on kappa receptors at dopaminergic neuron terminals. So dynorphin actually has the opposite effect of beta endorphin. Dynorphin can actually inhibit dopamine release onto the nucleus accumbens. So opioids can increase dopamine, but certain types of endogenous opioids like dynorphin can actually decrease release. Generally, what we're going to find is that chronic use is going to lead to sensitization and tolerance. So tolerance, as a reminder, is a diminishing drug effect with repeated use. And opioids, because they belong to similar classes with similar chemical structures, you can develop a cross tolerance. So if you have a really bad cough and you're a heroin addict, codeine is not really going to do a very good job of opening up those respiratory passages, because if you have developed a tolerance to heroin, you have developed a tolerance to other types of opioids. Sensitization is basically increased drug effect with repeated use. Now, whereas we develop a tolerance to a lot of the therapeutic or beneficial or enjoyable effects of opioids, cravings are going to actually increase with repeated use. So cravings will actually sensitize even as getting high off of opioids does not change, and it's even possible that cravings will sensitize even as the drug high decreases. Now, with repeated use, neuroadaptive changes result, which is going to lead to physical dependence. So basically, We're going to reduce our number of opioid receptors. The liver is going to develop enzymes that do a better job of breaking the drug down, and you're going to take more of that drug to have an effect. And this can lead to physical dependence. And this is particularly important for drugs like alcohol and opioids, because when the drug is no longer present, the cell functions are going to return to normal, and they're even going to overshoot. And these are going to be present in withdrawal symptoms, which will often be opposite of the effects of the drug. So withdrawal symptoms can vary based on a lot of different factors, like the drug that's used. For example, using morphine versus methadone, which is a longer lasting drug. Generally, drugs like methadone are going to have longer lasting symptoms, but they're going to be milder. So what you're going to see is that here are the acute actions of opioids, and then we'll talk about the withdrawal signs. And note that the withdrawal signs are often going to be opposite of the acute effects. So we have pain relief, respiratory depression, euphoria, relaxation and sleep, feeling tranquil, decreased blood pressure, constipation, pupil constriction, hypothermia, so being cold, drying of secretions, reduced sex drive, and flushed and warm skin. Here's the withdrawal effects. Pain and irritability. We're going to get more enhancement of respiration. We're going to have panting and yawning, dysphoria and depression, restlessness and insomnia, fearfulness and hostility, increased blood pressure, and it's this and the increased heart rate that really makes going cold turkey off of opioids dangerous. We have diarrhea, pupil dilation, hyperthermia, so body temperature being too high, tearing and a runny nose, spontaneous ejaculation, and chilliness and goose flesh. Now here, you can kind of see the intensity of the drug effects and the intensity of withdrawal. Generally, the more intense the drug is, the more intense the withdrawal is, but it's also going to be a lot more short-lived. So here's intravenous injection of heroin. You can see that those effects are almost immediate and they're gone within 8 hours. But those withdrawal effects are going to be pretty intense, but significantly more short-lived than methadone. Methadone peaks at about 4 hours of use, but you can see that it's still at a pretty high level, 28 to 32 hours after administration. Those withdrawal effects will also last a while. However, notice that they're going to be certainly not as intense as you might experience with something like heroin. So there's an inverse relationship between the intensity of the effect and how long that effect lasts. Now, what sort of things eliminate withdrawal symptoms? Well, generally, we do not want somebody to quit opioids cold turkey, much like we don't want somebody to quit alcohol cold turkey. Both of those cases can be really dangerous. They can lead to things like strokes and death, etc. So the goal initially is to detox. This is basically the point when the withdrawal symptoms end. And basically what we find, and this is where it gets really tricky, if the opioid is re-administered before that point, you get rid of the withdrawal symptoms. However, you have stopped detox. Now, we do know that there is a cross dependence with other opioids, and this can actually stop and reduce those withdrawal symptoms, which is why methadone is a really potentially promising treatment for a lot of people, because you are getting rid of those withdrawal symptoms, and because of that, people aren't seeking these drugs to aid their withdrawal symptoms. Now, you can have a dependence without an addiction. This is especially true for people who use opioids for chronic pain use. We do actually find that chronic pain patients do show signs of tolerance and dependence, but they're not necessarily addicted to it. So this is a case where practitioners have to pay careful attention to the dosage, and you need to reduce those doses over time. And with that, withdrawal symptoms and drug abuse are a lot less likely to happen as a result. So what parts of the brain are involved in withdrawal? When researchers have applied opioid antagonists to the brains of opioid-dependent animals, they found that areas like the locus coeruleus and the periaqueductal gray were the most sensitive to the antagonist. So the locus coeruleus and the periaqueductal gray seem to play a role. We also do find that the nucleus accumbens is important for the aversive effects and the motivational aspects of withdrawal. Now, here's what's kind of interesting. Most animals prefer novelty. But if you provide an opioid-dependent animal with an opioid antagonist to the nucleus accumbens, they actually end up developing an aversion to that novelty, which goes against how most animals tend to behave. So it looks like the locus coeruleus, the periaqueductal gray, and the nucleus accumbens are going to be really critical in those withdrawal symptoms. Now, I want to talk about, we already kind of talked about the opponent process theory of drug addiction last week, but another theory that has been involved with opioid withdrawal comes from Himmelsbach. And basically, Himmelsbach believes that the goal of the body and the brain is homeostasis. So basically, everything is perfectly balanced. But when you inject the presence of a drug, you are disturbing that homeostasis. And basically, what is going to happen is that your brain is going to develop compensatory mechanisms in the face of repeated administration of that drug to achieve new homeostasis. So the nervous system is going to basically adapt to that drug presence. That homeostasis is basically going to result in tolerance. If the drug is removed, one of the things that we know is that the way that your body acts to the drug is very fast acting, but the compensatory mechanisms that help re-achieve that homeostasis are a lot slower in response to change. So they're still going to be operating even if you stop taking that drug. And as a result, when the drug is removed, we are back at our disturbed homeostasis and we're totally running on those compensatory mechanisms. And as a result, we get that because we no longer have that drug, we're getting the full compensatory mechanism, we're getting that full rebound, and we're getting those withdrawal symptoms. Now, what is the evidence for this Himmelsbach model? So one of the things that we know is that morphine inhibits adenyl cyclase. This is an enzyme that basically makes cyclic AMP. So here we have an animal at normal. So basically it's making a full amount, 100% of cyclic AMP. And then we begin the morphine treatment. And what you can see almost immediately is that cyclic AMP drops, but eventually it returns to its normal rate. So we're kind of getting that homeostasis here. So to be able to produce the compensatory mechanisms are basically ramping up cyclic AMP production to basically counteract the reduction that the drug induces. When the morphine is basically withdrawn, at that point, you are totally operating on those compensatory mechanisms. And as a result, look at what happens. The cyclic AMP jumps to almost 400% of production. So withdrawal is associated with a significant rise in cyclic AMP levels, and then eventually, once that those compensatory mechanisms change, we return back to that normal rate. So there's some pretty strong evidence for the Himmelsbach theory of drug addiction and withdrawal. And conditioning also plays a role. One of the things that we find is that repeated administration in a given environment is associated with anticipatory physiological effects in animals. So for example, morphine slows down heart rate. When a rat goes into an area where it has consistently received morphine, it will start to slow down in anticipation of that morphine. And similar effects can lead with play a role in humans. One of the things that we know is that many people who use drugs tend to prefer them in certain types of environments. And we do develop compensatory mechanisms to those familiar environments. But we find that tolerance lowers in a novel environment. Those compensatory mechanisms haven't been conditioned in this environment. As a result, some people will take the same amount of a drug that they normally take, but they take it in a new place that they're not used to, and they end up overdosing because they don't have the conditioning to that environment in place. And so in addition to this, we know that there is secondary reinforcement of environmental factors. We can develop reinforcement through the social groups that we use drugs with, or these rituals that we may do to inject different types of opioids. Now, another thing that I'm going to show you, one of the things that we do find in PET scans of drug users, and to be clear, we're not doing this with heroin. This is with people who have a dependence on cocaine. We see increase in activation in areas like the amygdala, which is right here, and then areas like the anterior cingulate cortex. These areas are often going to be really critical for emotional memory and for our expectations. So here you have people that are addicted to cocaine. And they either watch a nature video or they watch a video of somebody basically involved with cocaine paraphernalia. And what you can see is that there is significantly greater metabolic activity in areas like the amygdala and in the anterior cingulate. Both of these areas have connections to the nucleus accumbens. So these kind of cues, like the environment or the paraphernalia associated with a specific drug, can basically act as a primer and enhance craving and drug-seeking behavior. So let's finish up by talking about the different types of treatments for opioid abuse. So treatment, anytime that we're talking about substance use, we're involving a biopsychosocial approach. Chronic drug use and addiction is a multi-factored problem, and that's also true for drugs like opioids. So treatments must consider the physiological effects, so the biology, the psychological state of the individual, and environmental factors. Now, like alcohol abuse, detoxing is always going to be your first step. So detoxing is often going to involve long-acting opioids like methadone. You're going to gradually decrease that dosage over a few days. So you're reducing withdrawal symptoms to a manageable level so that people don't go running back to that drug, because as we talked about with the opponent process model of addiction, oftentimes by the time that you are physically dependent on a drug, you are not taking that drug to get high. That high may not be there. You are probably taking that drug because the withdrawal symptoms have become so unmanageable. Now, another one that you can look at as a potential detox method is clonidine. Clonidine acts on noradrenergic autoreceptors. So it's basically helping norepinephrine cut its numbers. If you want to be really technical, it's an alpha-2 adrenergic agonist. And it reduces norepinephrine in the locus coeruleus. Now, norepinephrine is typically going to be inhibited by opioids, but during withdrawal, they increase their firing, so we get a lot of sympathetic nervous system symptoms. So if we can cut and reduce norepinephrine during detoxification, we can feel less of those withdrawal symptoms. The problem is clonidine doesn't relieve all of the withdrawal symptoms, and it has its own unpleasant side effects. Methadone, as I kind of mentioned, is a common treatment for addiction after detoxification. So we have what's called a methadone maintenance program. Basically, what you are doing is you are taking methadone once or twice a day and substituting that for your heroin use. And the reason that you would want to do this, methadone is generally doesn't bind to opioid receptors as readily. So it's not going to get you high. but it is going to be an opioid that kind of reduces those cravings and potentially reduces drug-seeking behavior. And as a result, it can free up your energy for more productive activities. And the cross-tolerance with heroin ultimately is going to reduce the likelihood of a relapse. Now, the thing about methadone maintenance is that methadone is still an opioid. If you take multiple pills, crush it, and inject it, it's going to get you high. So oftentimes what is recommended for methadone maintenance is a daily supervised oral administration. Now, it's not going to make you high. It really won't, but it's going to reduce those cravings and those withdrawal symptoms. Additionally, by taking it orally, it reduces the likelihood of running into an injection ritual, which can be highly reinforcing, and it's reducing the risk of disease because often you are using that same needle over and over again, and you're running the risk for diseases like things like hepatitis, HIV, or AIDS. Now, buprenorphine, which is another opioid, is another maintenance method. It's often going to be sold under the name Buprenex. Buprenex is a partial agonist with weaker effects, and it has a longer duration than methadone, so you don't have to go in every day. You only have to go in maybe once or twice a week. Buprenex is also available as Suboxone. So basically, we combine buprenorphine with naloxone. And the reason that, again, that you would want to do this is, again, Buprenex is an opioid. If you crush it and inject it, you're going to get high. If we sell it to you as Suboxone, naloxone is basically going to prevent the buprenorphine from getting you high if it's crushed and injected. But there's still potential for abuse here. Now, other treatments are going to use narcotic antagonists like naltrexone or Trexan. This has a longer duration of action than naloxone, so it's taken orally, very few side effects. There's nalmefene or Revex, very similar to naltrexone, but even more potent and longer lasting. But these drugs don't eliminate cravings. So it's really important to have a multidimensional approach. Yes, from the biological perspective, we do need to have detoxification, we need pharmacological support, and we need counseling. From the psychological, we need to identify environmental cues that really induce those cravings and come up with good behavioral responses when running into those cues or triggers. And something like Narconon is another option. So folks, that is all I had for talking about opioids. I will see you next time. We're going to talk about psychomotor stimulants.